So the work I'm going to talk about today is work that we have recently started doing in my team at Southampton. So I'm an associate professor. I did molecular genetics at Sussex a long time ago. I did some developmental biology and then actually got into paediatric cancer research at the Royal Marsden with a lady called Professor Janet Shipley, where I also met Robin Jones. And then I started working on adult sarcomas before I went down to Southampton to start my own team. And at Southampton, I met an upper GI surgeon who had been for many years resecting GIST cancers from patients. And this was material that had been collected, but there was nobody doing any sarcoma research at Southampton. So this felt like a great opportunity for us to be able to do something with all that really important patient material that we had at Southampton in the collection. So as part of a number of things that Tim Underwood, who is the GI surgeon who I will show you a picture of at the moment, one of the things we did was create a team. And what we're doing is slightly different to a lot of academic teams, because most academic teams are led by either a single clinician or a single scientist. But we decided that we were probably going to be more effective if we paired up and worked together. So we formed what we called the Innovation for Translation Research Group, and it is co-led by myself, a scientist, an academic scientist, and a surgeon. And we then, next slide please, we basically wanted to create a vision for our team that would encompass all the things that we do, which I'll go on to talk about, but I will very specifically talk about GIST. But we created a vision that encompasses essentially what we're trying to achieve within the lifetime of our team, which is really to make a difference to patients. So the work that we do is what we call very translational. So it's really trying to do something that we can move from the lab into the clinic as quickly as possible. So next slide please. Thank you. So we work on a lot of things in the team, as you can see here. So we do some stuff in adult pediatric diseases, some solid tumours and developmental disorders. Pediatric sarcomas is where I kind of made my name originally, but very recently, please ignore this pointer thing, it does this every time, but we've started working in GIST and esophageal adenocarcinomas. And as Nick mentioned at the beginning, we had funding from Sarcoma UK last year to do a little pilot project. And recently we've had the funding from Sarcoma UK and GIST Cancer UK to take a project further on, which I will talk a little bit about at the end. Next slide, please. So the way that we really want to try and make a difference is to, we can't do everything, right, because we're two people with quite a large team, as you'll see in a minute, but we wanted to really be clear about where we felt that we could best use our expertise and make a difference. So we're essentially involved in trying to understand what's going on biologically in response to therapy, and understanding things like treatment resistance, why is that happening and how can we overcome it? And also developing novel strategies to improve responses from the beginning, either in the resistance setting or in the kind of frontline setting. And because we work so closely, so Southampton University, I'm in the Faculty of Medicine, which is actually on the hospital site. So we have a really close relationship with the surgeons, the oncologists, the whole clinical team, which means that we can actually use all that information that we can get from patients like yourselves to really try and make a difference and benefit. So the next slide, please. So this is our team. And this guy in the middle is Tim Underwood, so he's the surgeon. And actually, I should say that we, again, another thing that we do slightly differently is actually these two ladies and this gentleman here are actually, so this lady has a mesenchymal chondrosarcoma. This lady, her son died of a spindle cell sarcoma. And there's another lady who's also had surgery. Tim actually operated on her. And we have what's called a strategic oversight team, where we actually involve patients and people related to patients in the work that we do so that the questions we ask are patient relevant. And actually that doesn't happen nearly often enough, and it's something we just started up this year, which is really exciting for us. So it means that we've basically got kind of patient oversight into the decisions we make in terms of what research we undertake and how we do it and that sort of thing. But if you could press the next one. So the guys I've highlighted in blue, are guys who are actually involved in the GIST project directly. So I mentioned Tim. This is another surgical registrar called Oliver Pickering, and he's involved in the project really heavily. This guy is a guy called Jack Harrington, who is a current PhD student, but who has done a little bit of the work so far, and who feels so passionately about GIST. He really wants to continue working in this field, so he's really keen to be a part of any work we do in the GIST field. And this guy is an oncologist called Luke Hill, he's trying to wrestle the GIST from the different, so it's quite complicated because we have a GI MDT and then we have a sarcoma MDT, and so we're trying to bring everything together so that the GIST sit in the right place and we can get all the information we need without having to move between MDTs and have all that complicated clinical crosstalk. And Luke is busy trying to just make sure he can kind of have control of everything so that we can really get the samples we need and the information we need to do something meaningful. And then down the side are Dean Bryant. So Dean Bryant is actually what we call the PI, so the lead for the new grant that's just been funded by Just Cancer UK. Zohor is a computer scientist who's going to be working with us. So Dean is what we call a bioinformatician. So he does computational work in the kind of biology sphere. Zohor is a bona fide proper computer scientist, so she's going to be helping with us. And this guy is a guy called Ben Sharp, who's helped us gather he was what's called a postdoc, so he did a PhD and then carried on doing some science with the team. And he, again, is also really keen to stay involved in the GIST projects. They are not, but don't they? Thank you very much. So now I'm going to get into the kind of technical science part, and the thing that we've really gotten interested in the last few years, along with many other scientific researchers and clinicians, is what we call the tumour microenvironment. And this is what we started to understand now is that tumour cells themselves don't exist alone, right? So if you go to the next slide, you have done it, thank you. So the tumour cells are represented here by these purple blobs, but what we're starting to understand now is that in a tumour mass, actually the tumour cells interact with what we call normal cells. So normal cells of such as what are called cancer-associated fibroblasts, so fibroblast cells, stromal cells which support the tumour, and also immune cells that actually will infiltrate the tumour and interact with the tumour cells. And there are scenarios in which this can be what we call favourable or unfavourable. So if you go to the next slide, what we're starting to understand now is that there are instances where tumours are what we call infiltrated, so mixed in with them are these immune cells, and actually immune hot mean that your tumours these immune cells are interacting and they are actually able to allow the tumour to be responsive to immune therapies, and actually these tumours tend to be responsive to immune therapies and have a better prognosis and that sort of thing because the immune system is able to recognise them as foreign. But on the other hand, what we now start to understand is there are instances where the tumour is able to signal, the tumour cells are able to signal to these immune cells and basically say, don't think that I'm foreign, don't recognise me, don't remove me, don't kill me. So they're basically, these cancer cells are able to evade the normal immune system. And when we're starting to understand now the importance of this in the prognosis of a cancer, how it's going to develop, whether it's going to metastasize, all of those sorts of things. So there's really key for us to understand not just what's going on with the cancer cells, but what's also going on with the other cells and the crosstalk between those cancer cells and those other cells within a tumour. Thank you very much. And so this is something that started to really sort of rear its head in the GIST cancer space. So I went to a thing called Fortress at the beginning of the year, which is a sarcoma meeting we have every year. And people are just starting to talk about this. What is the importance of the tumour microenvironment when it comes to GIST in all the different sort of senses of the word? So far, there's been very little done in this space. And the two published papers I'm just going to mention today, the only two published papers, use something that we call single-cell RNA sequencing. So to explain what that is and why it's important, historically, whenever we did some sequencing to really understand what the cancer cells were doing, so which genes were switched on, which ones were switched off, and what that meant for the cancer compared to normal tissue, we did something called bulk RNA sequencing, and that basically means that if you think about the context of that tumour microenvironment, you had all the tumour cells and all the normal cells all mixed up. And what you get out of it is a data a little bit like a smoothie. You don't know which signal has come from a tumour cell and which signals come from a normal cell, or how they might be talking to each other. What single cell RNA sequencing allows us to do is basically pick out your strawberries from your blueberries. So say, oh, that signal comes from a tumour cell, that signal comes from an immune cell. What does that mean? What are their networks like? How do they talk to each other? So hopefully that analogy helps explain this is exactly what happens. So this is a schematic of what we do within our lab. We take some tumour tissue. We do bulk RNA sequencing because it's cheaper and easier. But we also do single cell RNA sequencing, and now clever people like Dean can actually do what we call deconvolution of this. So if we have single cell data, we can use the bulk data to work out where the signals might have come from as well. But this is what we really need, is the single cell data to start off with, so we can work out what our tumour cells are doing, what all those normal cells that are mixed in with the tumour cells are doing, how they might be talking to each other. So the first bit of published data was from 2021, so a few years ago now. And essentially what they did was they took 2 patients with GIST, both KIT mutations. They then took a little bit of tumour tissue from the centre and a little bit from the edge, and did this single cell RNA sequencing. And what they found was, so I should say these 2 patients, both KIT mutant, one low risk, one high risk, was that actually there were a lot of immune cells in that GIST mass, both in the middle and at the periphery. So that means, technically speaking, GISTs are what we'd call immune hot, but they can be very aggressive. So what we need to know now is, if those immune cells are in there, why are they not doing their job? Why are they not attacking the tumour cells? But this first piece of work says there are immune cells in there, and actually there's no difference between what's in the middle and what's at the outer edge. So there's lots of immune cells in the middle and at the outer edge, right? But what are those cells doing, and why aren't they behaving like a normal immune cell and recognising the cancer cell as a foreign? And so that's probably the most important thing to take away from that paper. So literally a month ago, another paper came out, a second study from China, where they've looked actually and done this with 7 patients, imatinib sensitive versus resistant, and said, okay, what if we've got patients who are responding and patients who aren't? What's the difference in their tumour microenvironment, and what does that mean? So they've done the same thing. They basically took those patients, they did some single cell RNA sequencing, they did something else that I'll talk about later, and mapped the cells. A UMAP basically is this thing that tells you which cells are which. So basically, which are immune cells, which are tumour cells, that kind of thing. So it's just a computational methodology that computer scientists use. And the takeaway messages were that actually, again, there are large immune cell components, both in the sensitive and resistant. But what was particularly interesting is that in the resistant setting, they're particularly immunosuppressed. So actually, the immune cells in the imatinib resistance are really dampening down the immune system, which might be part of why some of these therapies aren't very effective, because there's no way for the immune system to work as well as the therapy working the way we expect it to. So this is quite interesting. But what's also interesting is there's, in terms of the different cell types, actually there was no difference between the sensitive and resistant. So they had all the same sorts of cell types, but actually what those cells were doing was slightly different in your 2 different settings. But that's all we know so far. So what kind of led us to this project is we had started doing our own single cell RNA sequencing. So we had done a couple of GIST patients, compared it to a normal gastric sample and leiomyoma, and this is basically just showing our data. So we had a little bit of data which we went to Sarcoma UK with and said, we really need to understand this, we need to understand, so the T up there is T cells, so that's immune cells, and again, like the other studies, you can see lots and lots of immune cells. So we again wanted to understand it, but what is missing from this data was the big question. So whilst we know that there are immune cells in there, And from that one study, we know that there are probably some in the centre and probably some at the edge. What we don't know is really where are they? How are they interacting with the tumour cells? Is it that the tumour cells are all together and the immune cells are surrounding them? Are they truly mixed together? Where are they sitting? Are they sitting on top of or below each other? Are they communicating with each other? So this is what we call positional information. So we know what cells are in there, but we just don't really know how they're talking to each other and what that means. We want to know where it is and isn't happening, if there is some crosstalk between the tumour cells and the normal cells. And what do those interactions mean? You know, for example, if a tumour cell is very near a T cell, one of the immune cells, is the tumour cell signalling to that immune cell to say, you don't recognise me. You don't recognise me as a foreign cell. Yeah. So, but we can't tell that from the information we've got so far. And the other thing is, everything that's been done so far is on what we call the mutant. So there's no information so far for the wild type SDH deficient. So this led us to our, what can we do? What is it that we've got in our sort of toolkit to help with this? So what we're going to use, and what we are using, so we've actually done our first one this week, which is very exciting, is something called spatial transcriptomics. Now this is where it gets super technical, but I will try and explain because it's beyond even me. I'm not a computational person. But what it means is that you can take what we call the H&E stain. So you can take the biopsy sample or your surgical resection sample, and you can work out exactly where your single cell signatures came from. So where are those cells positionally and which cells are next to them? So you take that information you've got from the single cell data, which is, you know, your immune cells, your tumour cells, your stromal cells, your other cells, but actually where is it coming from? So you can get that positional information and you can start to understand, are all the tumour cells in a cluster? Are they surrounded by immune cells? Are they infiltrated by them? Are there support stromal cells near them? And you can start to do lots of different things. You can also ask the question, okay, are the tumour cells that are near immune cells, do they look different to tumour cells that are not near immune cells, or tumour cells that are actually at the edge and things like that? So you can really start to pick apart what's going on in that tumour. And this is something that we've done really effectively with some other tumour types. So we've done this in an esophageal adenocarcinoma, and we thought, this is what's missing from GIST. This is what we want to do, because we can do this, let's do it. And so the first bit of funded work we've had from Sarcoma UK is to do exactly that. And we have, so at University Hospital Southampton, we have got 52 patient samples that we've got really well annotated clinical information for. We've got the mutational status, and we've also now, as of yesterday, signed an MTA with Royal Marsden. So we've also got access to the samples from the Royal Marsden, so a really nice big cohort that we can think about doing something with. I should also say as well, we've now got an MTA, so a material transfer agreement with the Val de Hebron as well. So there's a guy called Cesar Serrano at Val de Hebron who has a very large collection, and he's really keen to work with us as well. So we've got a really good collection of samples where we can ask these really important questions. So Sarcoma UK, about a year ago, funded this project that we call Defining the Tumour Microenvironment of GIST, with a view to identifying novel therapeutic strategies, because that's really the purpose for us. It's not just can we understand the biology, it's how can we leverage that biology, that understanding, to find something better in terms of treatment. So we're going to do this using what we call FFPE material. So that's just material that's taken either from a biopsy or from a surgical resection. This week we've done 2 kits, one PDGFR8 and a wild type, and actually an SDH deficient GIST. And our plan originally was to do 12, but we've developed a methodology now to do at least 24 with the same amount of money, which is great. So this just shows what you get off the data when you first do this. So this is the original sample, and these dots just show what it looks like to us when we put it on. So the dots are where we get bits of information from. And then you can map on what we call the gene expression at the end of it. So we can say, okay, basically, this is actually an esophageal adenocarcinoma, but if you've taken, a region here, what are these cells doing compared to this other region here that, you can quite clearly see looks quite different. So we can really start to understand what's going on at the gene expression level, what these cell types are, what they're doing, and how do these cells here communicate with these cells here, if you see what I mean. So we can start to do this. The difficulty is this is very challenging to analyse. And this has been one of those technologies where the technology has moved more quickly than perhaps the computational side of things, the ability to actually do the analysis. It's been a real challenge for our computational biologists. So unlike some of the older techniques, there's lots of things out there. People have developed these, what we call pipelines for analysis, that you can actually just use and apply. This doesn't really exist for spatial transcriptomics, which led us to the grant that has now just recently been funded by GIST Cancer UK. So this basically grant is a studentship to work with our computational scientists to come up with a really clever way to analyse this, so a brand new way to do it where we can get the most information from it. So essentially, so Dean made this slide for me, so the tech development has outpaced the ability to do the proper analysis and get meaningful information. So I've sort of talked about a kind of higher level of information, but there's so much more that can be done. And so really what we need is we need an analysis that brings together all the things actually that we do have at Southampton. We're really fortunate to be able to do that. There is a shortage of computational biologists and bioinformaticians, and this is a real problem. So this is why this funding is so important to us, is because we will be training kind of the next generation of a computational biologist, and we really need them given the amount of data that's being produced now. The University of Southampton has what we call a supercomputer. It's called Iridis. It's one of only 500 across the world, and it's a huge supercomputer that's really important in analysing all this data, because it is a lot of data. And it's a massive strength of ours at Southampton, and we have this access to these cutting-edge technologies. And so this studentship is going to basically allow us to train a new computational biologist who will then also be trained alongside our GIST oncologists and the GIST sarcoma experts at Southampton to really become a computational informatician who can really get to grips with this data and better understand. And the whole point really was to first of all establish the pipelines and really understand the data, generate these atlases to really understand it, and basically start to be able to understand things that will tell us when we might expect to see recurrence. So can we predict quite early where something might come back, where we might not have known or we might have thought that it wouldn't? And also, can we identify potential therapeutic targets? And this is a really big theme of our team. So we use computational data to be able to pick out new therapeutic targets quickly and easily. And we work with an AI drug discovery company, meaning that we can hopefully accelerate these things. So you saw the IDX study where they were talking about, this sort of 10 years. We're trying to kind of at least halve that pipeline, maybe shorten it as much as possible. And one of the things we do within our teams is find novel therapeutic targets and also try and look for opportunities for what we call repurposing. So can we find a target where there's already a drug in the market that we could give to a patient very quickly? So really trying to find things that can be rapidly moved, but also find new targets, work with AI drug discovery companies and try and really shorten that pipeline for something brand new. And the final thing we're going to do is validate our finding with something called multiplex immunohistochemistry. And this is basically, if you press the next one, this what you have to do when you do anything in a computational space and in a kind of big data space is then actually validate it. And multiplex immunohistochemistry, again, does something that we've only just really started to be able to do, which is look to see, okay, we predict from the RNA that this is present here and it's doing this particular thing. Can we now show it at the protein level? And multiplex immunohistochemistry is something, again, we've developed at Southampton, so we can stain for lots of different proteins and make sure that what we've found from our spatial transcriptomics and our single-cell data, and we've done all the computational stuff, actually stacks up when we go back to a large cohort of patient samples. So that's what we're going to do with the data. But that does leave us with one more thing that really we need to do. So I've just finished with a kind of what would we like to do next. So this is what we absolutely will do across the studentship, but what we would really like to do next is do some validation of findings. So we're collaborating with Jonathan Fletcher, Sebastian Bauer, Jason Sicklick and people as well. But what we would really like to do is to be able to move this forward, and what we're going to start to do and hopefully we'll be able to get some more funding to do this in a more meaningful way, but is to start to generate what we call patient-derived models of GIST. So this is one of the ways that we do this very effectively at Southampton, is to take the cancer material, so Tim, the surgeon, takes the tumour out of a patient, and then we literally take it back to the lab and try and grow those cells. And the advantage of these over what we call historic models, so cell lines, are that those tumours have come straight from the patient, and so they best represent the patient they came from at that moment, rather than a cell line that's been manipulated and changed and no longer bears much resemblance to the patient. So we've done this very effectively. We call it an organoid, and we've done this effectively for a number of sarcomas and for esophageal cancer, and we really want to try this for a GIST now. And thankfully, actually, Jason Siglick in the States just published a paper showing a protocol for how to do this. And it turns out it's exactly the same protocol as we've been using for our OAC. So we're really confident that the next GIST we get out of a patient, we're hoping we can start to make an organoid. What we'd really like to do in the future is do this particularly for the wild type in the SDH deficient, because there's a real issue there with any preclinical models. The reason we need what we call a preclinical model is so when we find that novel therapeutic target, we can test it and say, do we think this is actually going to work in a patient? And this is really what we would like to do next. That's it from me. Hopefully that was relatively clear, maybe as mud, but thank you. I will stay here.
So to me, these are the guys that when I was at school, you were in the biology lessons and really interested. Well, I was out on the sports fields playing sport. But I'd like to thank you both actually on behalf of us and the community, because when we first started doing these conferences where we had people come to give presentations, a lot of them were so scientific and we were sat there completely bamboozled by it. But I don't know if you agree, hopefully you'll agree with me, that both the presentations today have really brought it down to the sort of level that certainly we understand a lot more than we ever used to. Okay, there's some scientific stuff in there, but thank you for making that a lot easier for us to understand. It really does help. So, yeah. So, do we have any questions? Would you like to come up as well? So, I want to start actually with the first person. There's somebody at the back that wanted to ask a question. Just wait for me to get to you with the microphone.
There's a question for Dr. Bestan Thomas, please. You mentioned in your talk about the different pathways of all the different cancers. What's the sort of split between people who can go through surgery and people who can't go through surgery and go straight onto the medicine, the chemos, the imatinibs?
So I don't necessarily see the ones that go straight to surgery. So I never meet those patients. So all the ones I see have the sort of more advanced disease, either locally advanced or metastatic. But statistically, I think there's a high, the majority of people are eligible for surgery. But I don't know the ratio, I'm afraid. That's something I'd need to look into.
So we have the opposite. So at our place, actually, because I work with Tim, we are connected more to those who do go through surgery, and I think we must do sort of 20, 30 surgeries a year, maybe, but that includes any of the paediatric ones and things like that. So I think that's about right. I think most of them actually are what we call resectable.
Okay, any other questions? Yep.
I'm not sure who this is for, but can you tell me what the difference is between the single cell RNA and whole genome sequencing?
So whole genome sequencing is where you're sequencing the DNA. So in biology, your DNA encodes your genes. So your DNA is sort of the underlying thing that encodes genes. Now, your DNA does not change between your cells usually. So every cell in your body, a muscle cell, a neuron, blood cell, will all have the same DNA. Cancer cells not necessarily. So this is the point about the KIT mutation. That looks at the DNA level. So your tumour cell has a KIT mutation, but the rest of your cells may not have. Now, that's not always true. So that's not true of, say, SDH deficient, because every cell might have it, and that's called systemic. But the difference is that's about DNA, whole genome, whereas single cell is about what we call RNA. So what happens is your DNA encodes your genes, your DNA is transcribed into these things called called genes, and that's your RNA, and that's what we're measuring. So it's not measuring what's going on in the DNA, it's measuring, okay, which things are being switched on in which cells at which time. And the best way to think about that is your DNA is the same. So for, let's take a muscle cell and a neuron, a normal muscle and a normal neuron, they have the same DNA, but they're completely different, right? A muscle cell is a muscle cell and a neuron's a neuron. And it's the genes, it's which genes are switched on in those cells that dictate, am I a muscle cell? Am I a neuron? And actually that's also true of cancer cells. So the genes that are switched on can also say, actually I'm a cancer cell because I've got lots of genes, I'm producing lots of proteins that shouldn't be on. And that's really, so I'm a developmental biologist, and that's kind of my wheelhouse. And that's really what I sort of grew up studying, as it were, saying, okay, if there's not a mutation or there's not something about that cancer cell that makes it different at the DNA level, there must be something else. And it's this idea that actually your genes can be dictating that. Now the next level, I'm getting really complicated, but the mutations in the DNA can change which genes are on as well, and that's where you can look for some, what we call that therapeutic window. So it might be that you have a KIT mutation and it switches on a particular set of genes, and that's different to where you don't have one. And again, that gives us that opportunity to understand the difference between a mutant and a wild type, what things are switched on in the mutant, which things are switched on in the wild type, and can we find a therapeutic from that? And that's kind of it.
Mine is wild type.
Yeah.
Unknown mutation.
Yeah.
So I'm on my 3rd round of tumours. So they managed to do the second biopsy work, because they said they couldn't do it from bloods or anything else. It had to be from the actual tumour, and it can't be resected. So I just didn't quite understand the difference there between the two.
And wild type's a bit of a catch-all still, I think. And now that we can do, as Betsa mentioned, now that we can do whole genome, wild type doesn't mean wild type in the sense that we sort of grew up with it. It means non-KIT or non-PDGFRA, and actually it can mean there's something else there, but it doesn't have to. So I work a lot on paediatric cancers where they don't have any defining mutations. There's like nothing. And so we're like, well, it isn't a mutation-driven tumour. There's clearly something else, and that's where this idea of which genes are switched on where becomes really important. And what we focus on is saying what things are switched on in that cancer that aren't switched on in the normal tissues, so that we can try and find a drug to target those things, and that's really where we focus our energy.
Hi, my husband here is treated at Velindre under Dr. Cardiff Morgan. And he's SDHA deficient. They found out it was also hereditary, and our 3 daughters all have inherited the same gene, so they get MRI'd every 2 years. I know it's really off the wall, but I mean, how often does that happen?
So I actually took over the GIST practice from Dr. Cardiff Morgan, but I think she's kept some of the patients, and yourself included. But in the last 5 years, where I've been looking after patients with GIST, I've just seen one other with the SDH mutation that was genetic as well. So we do see it. But yes, as you know, it is very uncommon.
Sorry, as a side point, one of my daughters is she makes organoids for different types of cancer, and you've just come up with that actually, which is just really odd that she doesn't do it for GIST, but obviously I've just looked at that and thought that was really interesting as well.
That's wonderful,
so
yeah.
It's good to know.
I'm very happy to chat to her as well.
Okay.
Love to. Thank you. Love to.
I believe that Nick has arranged for me to speak to you guys after the meeting. But what I would like to say particularly to you, Zoe, is when I was at my last meeting, I was encouraged to get a sample of my GIST put down to the Royal Marsden. So after my operation, I convinced a very bewildered general surgeon that this was practical. So eventually he did. The coincidence of it all is I was travelling down here by train yesterday, and I got an email from the secretary who deals with this at the Royal Marsden asking me to update my consent form and initial it. So I checked what it was that was kind of different, and the bit that was that I would agree to third parties having access to my sample. And I have to say, you know, if I hadn't been here today, I would still be scratching my head and wondering, what does that mean? So I'm delighted. I may not have understood 100% of what you were saying. Your enthusiasm for the subject carried me right through. So I'm really glad that Just UK and you guys are all tied up together, and that I did put my sample down to the Marsden, so I know that you'll look out for it now.
Absolutely. Thank you so much. I won't
Thank
see
you.
it, but thank you so much. And it will make a huge difference to everything we do, especially in rare cancers. You know, I've worked in sarcoma a long time, and every sample is really key for us. It's really important. It helps us figure out how we can make a difference, what we can do. Just to give a quick example, we've been doing this in lipos for a few years, liposarcomas, and now we think we found a drug. So just in 2 years of doing a project with some computational data, doing a little bit of stuff in the lab, we're working with a guy in Belgium to see if we can test this drug in their mouse models and get that into a clinical trial. So we're really hoping we can do something that rapidly for GIST, but every sample is key in something so rare. So thank you so much.
So just to give some meat on those bones for those that don't know, so GIST Cancer UK set up a tumour biobank effectively, and it's now held at the Royal Marsden by Eniola. Is that who contacted you? Yes. Yeah. So she's taken it over. So if the only tumour samples we don't need is exon 11, because we've got hundreds of them. But any other samples, as has just been explained here, if you have your tumour removed, please by all means ask your surgeon if they can send a sample of it. Now it needs to be frozen, I believe, as opposed, or no? Okay.
No, so it depends what we're doing. So I think the easiest thing for us to do this particular piece of work is actually the FFPE, so it's not the frozen. When we're doing the single cell work, we have to take that straight from surgery. So it's sort of easier for us to do that at whoever's undergoing surgery at Southampton, whereas the spatial transcriptomics, again, that's what makes it so powerful, is we can use all those historic samples that have come through as FFPE. So it doesn't have to be frozen. And in some ways, it doesn't matter, because whatever was done to the sample, we can figure out a different technology that we'll use to do that validation part if we don't do the spatial. So I would say, do whatever makes sense for whoever's doing the surgery to get that important sample to wherever it needs to go to, because we will find a way to use it, whether it's frozen, FFP, or whatever it is, so that we can make the most of it.
And Eniola will deal with everything for you. You just need to contact her at the Royal Marsden. She will deal with the transport, the signatures that are required, etc. Get it into the biobank. So, question here.
Hi, it's about the mutations, and I've been looking into the sort of, insertion, deletion, substitute. Is there any research into sort of prognosis depending on which type of mutation you have?
Yes, there definitely is. And I listened to a few of the presentations that is on the website for the Just UK, so previous presentations, because this is relatively new to me, and I've certainly never spoken in one of these meetings before. But if you listen to previous meetings, I know Ramesh Balusu, for example, has done a talk on the different types of whether, yes, is it within exon 11, but it's a deletion or a mutation or a substitution. And there were some graphs in there that can tell you exactly, the chances of responding to a TKI depending on that particular type of mutation.
Okay, we've probably got time for one more question.
Can I just add to that really quickly?
Yeah,
Sorry.
sure.
For the first time this year, so Jonathan Fletcher and Sebastian Bauer are organising a GIST summit in October, and there are definitely plans to really try to bring together worldwide data to better understand that, because the sample set is so very small. It's very difficult to really make much of those where your sample number's small. So there is a real effort now across particularly Europe and America, so worldwide, to get all the data we can together to better answer exact questions like that. So that'll be really exciting.
So on that note, again, other things that we do is Just Cancer UK. So next week in Rome is the Spain conference. It's SPAGN, Sarcoma Patient Advocacy Global Network, where all the specialists in GIST come together. You're not, are you going? No. But it's a worldwide thing. They do one in Europe and one in the USA every year. I'm going as a representative of GIST Cancer UK to do that networking, to understand a bit more about what's going on globally within GIST Cancer, and I'll report back to the Board of Trustees. It's just another thing that we do as a cancer charity that maybe you guys don't see behind the scenes. So that's the other side. We were also going to Detroit, I believe, are we, Mark? Is that where you're going? Or somewhere in America. Oh, San Diego. Oh, there you go. San Diego for another worldwide GIST where all the specialists come together and we can go and annoy them and find out what they're doing. Can we help? Can we do anything to help you with your research that you're doing? You know, so all these little tiny things all help for the overall finding treatments and cures for GIST. So a last question here, and are you both staying for some lunch, or have you got a...
I could stay a little while. And then
Yeah, so there'll be time to have a chat for some questions anyway. So,
just a quick question. In the news, it's been mentioning that cancer can be detected in blood tests. Is GIST one of those?
Yes. So one of the slides I had was talking about the target trial that we're doing in Velindre, but it would be available in other clinical trials units or cancer centres across the UK, and that's looking at circulating tumour DNA, so that would be a blood test. So we're not routinely accessing that in GIST at the moment on the NHS, but in certain circumstances it can be accessed, and it's certainly something that you can detect. You can detect a GIST cancer in a blood test.
thank you again, both of you.